Okay, so we are back to our discussion of restriction fragment length polymorphism. So this kind of experiments are basically used for DNA fingerprinting and also to detect some kind of diseases. So in this particular PDF file of our experiment that we are going to conduct in our lab, they have also given an example of a disease, like how exactly you can go ahead and use this particular method, molecular diagnostic method, to detect the genetic disease. Okay, so we'll go through it and then I can explain you the experiment in short or in detail, however we go about it. So Rflp analysis was the first DNA profiling technique for genetic fingerprinting, genome mapping, localization of genes for genetic disease, ठीक है, disorders. So determination of risk of disease and paternity testing. So these are all applications of Rflp that we can use. Presence and absence of fragments resulting from changes in recognition sites are used for identification of species or population. So presence and absence of fragments resulting from changes in recognition site. ठीक है? So one of the DNAs, if you have two samples of DNA, you can have different means the EcoR1, the restriction enzyme will detect one particular sequences only. But it is not necessary that all the DNA samples will have that particular recognition site on them for EcoR1, any restriction enzyme to work, right? Because we all know that for restriction enzymes to work, they need to have a restriction site, right? Recognition site, I mean to say, not restriction site. They need to have a recognition site on that DNA. Only then they can go ahead, detect that recognition site, and then only they can cut the DNA into fragments, right? So depending on that, so when I have two different samples of DNA, it is not necessary that both the samples will have same kind of recognition. Means like same, they might have a recognition site, they might not have a recognition site. So depending on that, where the restriction enzymes are finding the recognition site and cleaving the DNA, we'll be getting different fragments of DNA, right? So accordingly, we'll be getting a profile for each DNA, understanding this part. So when you want to differentiate between a DNA of a mother, father also, sorry, a child and the father and child and the mother, whatever you want to do, you can choose this method and you can look for the similarities. For the forensic application also, you can go ahead and do the same. I have explained you about the forensic application before also. You can get the DA, get some sample from the crime site, and then you can analyze the DNA, you can do the DNA fingerprinting for that crime site sample DNA and suspect DNA, and accordingly you can find out whether the person we are suspecting was really present at the crime site or not, or he is the one to be blamed for any kind of a crime that has happened, right? So there are many applications of Rflp in forensic area, forensic science also. Then we read this particular application of Rflp that they have given us. So application of Rflp in mapping genetic disease, very interesting one this is. So for the detection of sickle cell anemia, DNA, so sickle cell anemia, everybody must know that it is the disease of hemoglobin when you do not, there is a mutation in your hemoglobin gene, okay, or you have a different allele that is present, okay. So the DNA from the hemoglobin gene from each family member is digested with particular restriction enzyme. So in a family, if you want to know who all are the carriers of this sickle cell anemia disease because it's a genetic disorder, or who is having sickle cell anemia, or who is not having sickle cell anemia, you can go ahead and extract the DNA from the blood of each member of the family. And since the hemoglobin gene is polymorphic, So when you say, what do you mean by polymorphic? That it can exist in two different forms on the same locus. So when I'm saying locus is the place where that particular gene is present on the chromosome. मीन्स वो कौन से जगह पे आता है क्रोमोसोम में, उस जगह को बोलते हैं लोकस. ठीक है. So when a person is having sickle cell anemia, he will have the hemoglobin gene, but it will be a different form of hemoglobin gene. So hence it is called as polymorphic, in the sense the hemoglobin A is the wild type, in means that is the one which will regulate for normal hemoglobin production. But HbS is what? It's an allele which is defective in nature. Okay? So it has a little bit of a difference than that of HbA, which is a wild type, and but it is present at the same position as that of HbA on the chromosome, right? So instead of having wild type HbA, the person will be having, the one who is suffering from sickle cell anemia will be having HbS type of the hemoglobin gene on the chromosome. Okay? So since the hemoglobin gene is polymorphic, there is more than one DNA sequence encoding for the gene. Okay? Means this one can also encode for the hemoglobin, and this one can also encode for hemoglobin. But you see, this one will be a defective one, right? So HbA is the wild type allele, and HbS is the allele that codes for sickling of red blood cells, right? So HbA is the wild type, means that is you and you, me, all of us, we have a wild type hemoglobin gene. Okay, wild type hemoglobin allele, that is HbA. But the person who will be suffering from sickle cell anemia will be having HbS, which will code for sickling of the red blood cells. So Rflp are produced using this polymorphic DNA sequence, and resulting fragments are separated by agarose gel electrophoresis, and as shown in the figure. So what they will be doing is, in order to find out whether that person is suffering from a genetic disease as that of sickle cell anemia, they'll be taking samples from each member of the family. Okay? Because it's a genetic disorder that will be carried over from mother, father to the children, and so on to the next generation. So they can take the blood samples from the family members and extract the DNA and go for the Rflp procedure to find out whether which of the individual is actually suffering from sickle cell anemia, or which of the individuals are carriers of the sickle cell anemia, and which of the individuals are wild type for hemoglobin gene, right? Means they are not having any traces of sickle cell anemia. So why do you need to do that? See, now we know that sickle cell anemia is a genetic disease, right? So if we have a history of sickle cell anemia in somebody's family, so if you want to protect the newborn individual, whether you want to know that the newborn is going to have the sickle cell anemia or not, you would want to do the genetic mapping beforehand only, right? Rather than having that individual come into the life and suffering later on, you can do the genetic mapping beforehand, and then you can come to know whether the new child that is going to be born will be a carrier, or will be a normal child, or will be suffered by sickle cell anemia. So that could be an advantage of having this particular Rflp test done on the genetic disorders like sickle cell anemia, because that is how you can prevent these genetic disorders from transferring to the this thing. So all of us, we know Mendelian genetics. We all know that how the disease is transferred from one generation to another generation, depending on whether the mother is the carrier, whether the father is the carrier, and how it will be getting into. Not all the children will be affected by sickle cell anemia, but one of the child could be affected by the sickle cell anemia, and this is what a pedigree that we can draw out of by knowing that who all suffered by sickle cell anemia in the family. Okay? So what is the main thing over here that I want to tell you is like how exactly this particular disease is detected with the help of Rflp. So you can see over here, this is the wild type hemoglobin gene, and this is the one which is a mutated allele. You can call it as a mutated allele. And you see the restriction sites are cutting at this particular site, 1, 2, and 3. So these are the restriction site locations. So these are the recognition sites for the restriction enzymes. And over here you see for HBS, this particular site is missing, you see, and hence the DNA fragment that is forming is 1.35 kilobytes, the kilo base pair, sorry, not bytes. So it's not computer. So and over here it is 1.15 kilo base pairs. So if you see now this Rflp pattern for the detection of sickle cell anemia. So if you have an individual who is having both the alleles of HBS and means who are heterozygous in nature, so they will be having a band at 1.15 also and 1.35 also, but they will not be suffering with the disease because they have the normal hemoglobin gene as well, right? Because they also have a wild type HbA, hence they will not be the one who will be suffering from the disease, but they could be the carriers. Same is the case for this. But if you see this one, this will be a wild type one because it has only HBS, HBAA gene present, allele present on that particular locus of the gene, a chromosome. And over here you see SS is the one who is showing the fragment only at 1.35 kilobase pairs, right? So this is the individual who is going to suffer from sickle cell anemia. Okay? And these ones are again the heterozygous one who could be the carriers. Okay? So what do I want to tell you over here is that, see, when you have a wild type, you will get the fragment, DNA fragment of 1.15 kilo base pairs only. Okay? When you have a wild type. So when you have both the alleles present, you will get the DNA fragments of both the lengths, that is 1.35 kilobase pairs and 1.15 kilobase pairs. But if the person is suffering from the sickle cell anemia, you will get the fragment only of 1.35 kilobase pairs. And that is how you can easily identify a person who is having sickle cell anemia and who is the carrier of the sickle cell anemia, and a person who is not suffering from the sickle cell anemia in a family. So the wild type hemoglobin gene, that is HbA, is shown a band at 1.15 kilobase pair, while the sickled hemoglobin gene, that is HbS, shows a band at 1.35 kilobase pairs. So a person homozygous for sickle cell anemia, that is SS, shows only one Rflp at 1 restriction fragment length polymorphism at 1.35 kilobase pair, while people who are heterozygous for this disease AS have Rflps at 1.35 and 1.15 kilobase pairs. So people who cannot inherit this gene, that is AA, show in the Rflp having the band only at 1.15 kilobase pair. Therefore, by studying the Rflp pattern, one can detect the presence of genetic disease in a certain individual, right? So, if you know that this certain individual will be affected by sickle cell anemia, you can take necessary precautions, because for sickle cell anemia, the only treatment you have right now is blood transfusion. You will constantly keep on changing the RBCs, replacing them with the new RBCs who will not get sickled because their gene is only defected. So when you have gene which is defected, there is no way you can protect those RBCs from getting sickled, right? So everybody must know the meaning of sickled, right? What do you mean by sickled? They will be like this. So RBCs are like concave, must round wala structure, right? These are our normal RBCs. So sickled in the sense they become moon shaped like this, right? And this is when they cannot absorb lots and lots of oxygen, and hence it will create lots of problem in your body system. Understood? So if you still have any questions about what is sickle cell anemia, we have YouTubes, we have very important many articles also on Google. You can look for any article and learn about this disease. But my goal is to was to tell you that how we can use this Rflp to determine so many genetic diseases, and also in forensic sciences, and also for paternity, maternity test, etc. So many things are there. So I hope you guys have understood this particular concept of RFLP for detection, application of RFLP for detection of genetic disease. Yes or no? Have you guys understood it or not? You can type me in the chat box. Yes, very simple one to understand. See, that's the reason, yes, that's the reason I was very keen about making you guys learn about the restriction enzymes. See, many of the micro and biotech students might know what are restriction enzymes are, but for chemistry people, it's a different knowledge, it's a new knowledge. So, why these restriction enzymes are important and how we can utilize these restriction enzymes, that is what is the important thing. Okay. So, yes. So moving on, so what all we have in this kit? So we have some reference samples of DNA. So we will be given one sample, test sample, and we'll be given three reference samples. Okay? And will be provided with the restriction enzymes. So what you will have to do? You'll have to treat these four samples of DNA. When I'm saying four is what? Reference sample 1, reference sample 2, reference sample 3, and your test sample of DNA. You'll be treating these four samples of DNA with the restriction enzymes. And which are the restriction enzymes that are we using over here? EcoR1 and PST1. Okay, so these are the restriction enzymes that are we going to use, and we are going to treat equally all these four samples. We are going to treat them equally with these two restriction enzymes, and we'll be getting different fragments of DNA when we treat these DNA samples with these restriction enzymes, and we'll be getting different lengths of DNA, different fragments of DNA, which will be running on the agarose gel, and then we'll be checking the DNA profile of each DNA of our sample DNA of the reference DNA of ours, and we'll be matching those fingerprints. Okay, so how it is done, I will let you know. How I will tell you how it is. So basically we have over here is that we'll be making four reaction tubes, and in each reaction tube you will be adding a sample, DNA sample, 10x assay buffer. When we say assay buffer, it will have nothing but T, right, to maintain the pH so that DNA is not degraded, and then it will have what you say, maybe magnesium ions for the stability of the DNA. So all these things will be there, which will make sure the stability of the DNA, and there is no degradation of the DNA throughout the experiment. And so we'll be adding 15 microliter of DNA sample, 10x acid buffer of 3 microliter, then molecular biology grade water 10 microliter, then eco R1 and PST. When you see the amount of restriction enzymes that we are adding, the amount of restriction enzyme is so less. It is what? 1.0 microliter, and this one also 1.0 microliter. That is total 30 microliter of the reaction mix you will be having. See, this is a very critical experiment, and it needs very uh, matured technical skills for you to perform this experiment, because most of the time क्या होता है, मीन्स हमारे लैब में ये बहुत बार हो चुका है कि 1 माइक्रोलीटर लेना है हमें, ठीक है, 1 माइक्रोलीटर ऑफ रेस्ट्रिक्शन एंजाइम, and what happens is like people are in the queue waiting for their turn to come to make this reaction mixture, and because of the bad skills of one of the person, instead of taking 1 microliter of restriction enzyme, they end up taking 4 microliter of restriction enzyme, because we all know that we have to stop at first knob and then take while using the micropipette, you have to stop at the first knob, then you have to suck the solution, and then accordingly you'll have to dispense it into your tube, reaction tube. But people will end up taking 2 or 3 microliter by going deep into that knob and then taking the solution from this particular cartridge of ours. And by the time we reach to the 10th person, we are finished with the restriction enzyme. So that is the reason I always sit with my students for performing these experiments, and I have to let them know that how do the 1 microliter looks like in that particular tip of micro pipette. So very crucial experiment, very interesting experiment. You really go through the experiment and wait till you see the results. So this is a typical reaction that we'll be having. We'll be setting it up and we'll be incubating this particular reaction mixture at 37 degrees centigrade for 2 to 3 hours. So we'll be putting them into the incubator. And after the incubation is done, we have to simply run this on the agarose gel electrophoresis. So once you run them on the agarose gel electrophoresis, you will be getting a pattern something like this. So this is your molecular weight DNA marker, and this is reference sample 1, reference sample 2, reference sample 3, and you will be having one test sample over here. Okay, let me show. So you'll be having the gel run for reference sample, your test sample. Okay, so test sample. Okay, and you will be getting profile like this maybe. Okay, and then you can match the fingerprints of your test sample with your reference samples by counting the molecular weight of the DNA fragments that you have achieved. Okay? So most of the time in our experiments, we can just look at the results that we have got under the UV illuminator, and we can tell that whether we have the test sample is matching with which reference sample. The DNA fingerprint of the test sample is matching with which reference sample. But ideal way of calculating and matching this DNA fingerprinting profile is by determining the molecular weight of each DNA fragment that you have got. And determining the molecular weight of the each DNA fragment is same as that of how we determine the protein part. If you guys remember the amount of distance traveled by amount of distance traveled by the band, and dividing that and plotting a graph and extrapolating the stuff, and you will be knowing the weight of each DNA fragment of the test sample, and you'll have to do the same for the all reference samples. And by making the table format of all the DNA fragments and their molecular weight, you can compare it accordingly. Okay. So nowadays we have gel docs available which will directly calculate the DNA fragments molecular weight with the reference of your DNA ladder, and will give you the results directly. But that is the advanced technology. So if we teach you directly how to use the software for calculation of this DNA fragments length, like their weight, then you guys will never understand the funda behind how exactly the calculation is been done. So this is how we perform this experiment of restriction fragment length polymorphism. So basically in the experiment you have 3 reference DNA sample, 1 test DNA sample, and you will be treating this restriction this DNA sample with same restriction enzyme, which in this case are EcoR1 and PST1, and you will be checking for the fragment profile, DNA fingerprint profile. Okay, DNA fingerprint, and then accordingly you will be making the conclusion. So from all the restriction profile obtained on, one can observe that restriction profile of given unknown sample matches with which reference sample. So you have to compare like which one will be matching with the reference samples, and as the restriction enzymes recognize and digest a particular sequence, any side change in that result in different restriction profile of the particular sample. So main thing that I want to tell you guys is that we keeping the restriction enzyme same is to what, that we want to make each DNA to be treated in the same manner as that of means all the DNA test DNA. ठीक है, तो तुम्हारा रेस्ट्रिक्शन एंजाइम सेम ही होना चाहिए, ठीक है। सो इन ऑर्डर टू नो, इन ऑर्डर टू गेट सम अप्रोप्रिएट रिजल्ट्स, ठीक है। सो ऐसा भी नहीं है कि तुम कौन सा मैंने एक प्लांट का डीएनए सैंपल लिया है, एक एनिमल का डीएनए सैंपल लिया है, एंड देन आई एम कंपेयरिंग, दैट इज नॉट हाउ यू कंपेयर, right? So how you will be comparing is like how we compared like wild type with the mutated. So if I have wild type gene and I have 2 3 samples of DNA which I have collected from few people who are having some particular type of a cancer or something like that. So I can then compare the DNA of those people with the wild type DNA, and then it's like the wild type of the normal DNA, and then I can have a profile in front of me, and maybe I can take my research further from that point onwards. So that is also, so you have to think logically, you have to think innovate in a innovative way, so that you can come up with your own protocol. So that is what it is nowadays. Molecular biology is all about having your own protocols made, having your own knowledge being implemented at that particular molecular level, because you know, all we all know that in this corona research also, COVID-19 research also, people are being innovative in so many ways, right? Finding that Arogya Setu app, finding of how will you go to how can you go about tracking people. Innovation is there also, right? Korea ended up tracking people by tracking their mobile phones, whether they are quarantining themselves or but. So that is innovative way of doing things. So innovation is something that you will be doing in order to reduce the work of a human being, which you can perform it in a very less amount of time, ठीक है। So ये सब करने को what I want to tell you is like to perform this RFLP experiment, right now we saw the incubation period is of 3 hours, ठीक है। उसके बाद हम जेल इलेक्ट्रोफोरेसिस करेंगे, फिर उसके बाद वो रन होगा। तो ये पूरे एक्सपेरिमेंट को we literally spend the whole day, right? So what could be the edge through in this is to have an experiment which can be run within an hour, right? And that is how in COVID-19 also we started from PCR and now we are at the antigen testing kits, right? So that is how the research goes, and that is how your innovations come into picture, and that is how we make the work easy for all the human beings. So but these are the basic ideas with which you have to work and play around with, so that you can have some different things popping up in your mind, okay? So this was about RFLP. Are there any doubts in this? As we proceed, as we do the experiments in our lab, we'll be understanding it. And since it is related to the topics that I'm teaching you right now, restriction enzyme, etc., so I thought it will be better to discuss the experiment also at the same time, right? So are there any doubts in this? You can write yes or no. Tanuja, are we fine till here? I'm worried about the chemistry people. Are we fine till here, Tanuja? Tanuja, Samruddhi, Shraddha, you guys have to let me know. Supriya, because I'm sure the biotech and micro people are pretty much fine with it. Okay, okay. So yes. So this was about RFLP, and then we have to start with a new topic, but we have 5 minutes. Maybe I can give you the introduction of this. So we are now at the point of learning about vectors. Okay. So vectors are generally used for gene cloning. Okay. So we'll start with the definition of gene cloning. What do you mean by gene cloning? So the clone from Greek word is clone is a twig. Okay. So the production of exact copies, that is, those will be called as clones of a particular gene or DNA sequence using genetic engineering technique will be called as what? Gene cloning. So the production of exact copies of a particular gene or the DNA sequence using genetic engineering technique. So the DNA containing the target genes is split into the fragments using restriction enzymes, and these fragments are then inserted into the cloning vectors. Okay, so you will be fragmenting the DNA using restriction enzyme. We all know very well by now what are restriction enzymes, and these fragments are then inserted into cloning vectors. So cloning vectors. So these are what? These are the medias who are going to have our DNA of interest. So they are going to be the vehicle. Vectors are going to be the vehicle who will be carrying our DNA of interest, which transfer the recombinant DNA to the suitable host. So vectors have our DNA of interest, and these vectors, which then will be called as recombinant DNA, will be transferred to a suitable host for making the multiple copies of that recombinant DNA. So inside the host cell, the recombinant DNA undergoes the replication, and thus the bacterial host will give rise to colonies of cell containing the cloned target gene. Okay. So there are many things when we will be learning about vector, the how vectors should be, what are the properties of the vectors, and then when you are going to select a vector, basically when you are going to select a vehicle for adding our gene of interest, that is DNA of interest in that particular vector या फिर vehicle, how exactly that vector should be, that is what is most important part in gene cloning, because how you are selecting your vector, depending on that only you will be getting the copies of recombinant DNA. And you have one experiment related to this also in your fourth semester, which is known as blue and white screening, in which we'll be making recombinant DNA and inserting that recombinant DNA into the plasmid of bacteria, and then allowing that plasmid to be inserted back into the host bacteria and multiply, making sure that we are getting the copies of those bacteria which are already which are having that plasmid, which is having our DNA of interest. So you have recombinant DNA will be inserting into the bacteria and multiplying those bacteria, right? So that is the main goal of blue and white screening. That is nothing but your transformation experiment, right? Transformation of the bacteria. Transformation is what? In which you will be adding the recombinant DNA, that is your recombinant DNA, which is like plasmid and the DNA of interest, fragment of DNA of interest, and you'll be inserting that back into the bacteria and allowing only those bacteria to grow who are having our recombinant DNA. Okay, so that is what your transformation experiment is all about. Then, what are the significance of the gene cloning? So, a particular gene can be isolated and its nucleotide sequence can be determined. Control sequence of DNA can be identified and analyzed. Protein, enzyme, and RNA functions can be investigated. Mutations can be identified, such as gene defect related to specific disease, and an organism can be engineered for a specific purposes. So these are all the significances of the gene cloning. Okay. So we have less than 1 minute remaining. We'll be continuing this topic tomorrow. Okay. I hope you guys are understanding and you guys are with me when we are doing all these readings and discussing the stuff. I understand it is really one way, but this is how it is when it is about online sessions. So if there are any doubts, you can post it to me, you can call me, you can WhatsApp me, you can always speak up during the class also. So I hope you guys are doing well by far. And I'm signing off for now. Thank you for being here. Please attend the next lecture as well. Okay. See you all.